So we're 30 minutes into our podcast, and I know the rule of thumb is podcasts should only be about 20 minutes, right? But I wanted you to have some of these basics. We'll go through some very basic, we might speed it up, and then we're going to take it back to nursing and where we're going, okay? So let's let the review of the microbiology and G&G boot camp begin. So we talked about the Human Genome Project, right? And the first sequencing of human DNA, and I gave some information there. We're also going to talk about actual definitions, right? And what is it? And genetics is the study of traits and conditions that are encoded in block sequences in the molecule of DNA. I'm going to show you a few little maps and diagrams that are going to help. But again, what does it really give you? What does your DNA give you? Your ancestry. You might find some siblings or relatives that you didn't know. There's a lot of things that are within there, okay? And we talked a lot more about variants, risks for cancers, and then we talked about pharmacogenetics as well. So, again, we always bring everything back to movies and things like that, right? How are we related? How is heredity a concern when it comes between generations and transmission of traits? And so again, that's what it's all about. I want to talk a little bit too about what makes a cell different from another, and you really see what these terms are. You saw this a lot in microbiology. This is just to kind of clear out the cobwebs, right? And we've got the genome, we've got the chromosome, the DNA chain, and genes within that. Again, more vocabulary. We have about 20,000 to 30,000 genes, right? And again, we'll talk more about some of these things as we move along. The human genome contains about 3 billion base pairs or nucleotides. Again, just a feedback back to our microbiology. Again, more structure pictures, and to show you that each nucleotide base pair has the GCAT within it, right? The guanine, the cytosine, the adenine, and the thymine aspects. And I think you'll remember that. And I've broken it down a little bit more, components of phosphate, sugar, and base. And here they are of how they kind of bind together, right? The chemical structure gives two key abilities essential for being the basis of life. It can replicate itself when a cell divides, and it holds information, right? And again, a little bit more information, right? Of what exactly the DNA, the RNA, protein, and the cells and tissue, right? It's a recipe book, right? I have a little information here, and later when you get the PowerPoint, you can play this little TikTok kind of thing here too that really talks about replication, transcription, the gene expression then and the translation. So that is just another feedback as well. And it may start when I click this.
This is arguably one of the most important aspects of biology that you should know. It's called the central dogma of molecular biology. A dogma is like a set of principles, and like in this case, that all of molecular biology follows. There are 3 steps to the central dogma of molecular biology. In order they go replication, transcription, translation. Replication is when DNA copies itself, shown here in our illustration. Transcription occurs when DNA copies itself into mRNA. Translation occurs when this mRNA sequence is translated into a protein. Both replication and transcription are going to occur in the nucleus because they involve our DNA. Translation, however, occurs at the ribosome because that's where we produce our proteins.
So there's a little review for you on that. So if you're an X-Men and all those kind of things fan, you will really there's a lot about mutations, right? Within comic books, within the movies. And so a change in a gene is a mutation, and it can have an effect at the whole person level. Alleles are the variants of genes. So they can be inherited or arise by mutations. Again, there's a lot of information there. Mutations in sperm or egg cells are passed on to the next generation. And remember, these can be positive, negative, or neutral. And that's the important things. Mutations have a connotation that they're always negative, but that is not so. So we've already talked, and again, going back to some movies, we've already talked about mutations that can be DNA or they can be environmental, right? And again, you can see the mutant turtles there. You can see environment, right, through insects. And then you see from the movie Wonder, the craniofacial newborn condition that the particular character had. So you can see that, what one chromosome, sort of the different aspects of that. And I won't go through that. I'll let you look at that. But again, it shows you sort of the characteristics of the chromosome. So again, just to give a little bit of information, it shows you that, a little bit more about tissue cells, how many cells we have in our body, and that tissues are groups of cells that share a function. Again, we look and say that every cell in our body has the same DNA, okay? And then there's the karyotype, and we'll talk a little bit more about karyotypes and autosomes and sex chromosomes in just a second. So, your karyotype is the individual's complete set of chromosomes. Having said that, we have 23 pairs, right? And 22 are autosomes, and the 23rd pair are sex chromosomes. And again, there's a diagram of that. This is a picture too from I've created a Jeopardy game to kind of test yourself. And we probably won't have time to do it in class, but I've given you that source that you will be able to do that. But it'll kind of quiz you on some of the things that we're talking about now. From this Jeopardy game is also another thing. I put a question in there that says, this percentage of DNA sequence is estimated to be unique in individuals. And as you can see, technically, we are 99.9% identical. We have a possibly 0.1 to 0.5% of differences. And I think when you look at that, it's sort of, it kind of shocks us, right? In genetics and genomics, we will say that Mendel is sort of our discovery of traits, right? And what are traits? And again, I gave you that information here. We also remember doing Punnett squares, right? Making these dominant and recessive abilities in our family history. What is considered dominant? What is considered recessive? We've talked about pedigrees already too, and genotypes and phenotypes. There's definitions there. And how do we sort of look at the likelihood that someone will have a certain condition or a certain genotype or phenotype? Along with that, I pose a question here. If one parent is heterozygous for brown eyes and the other parent is homozygous for blue eyes, what's the chance they will have a brown-eyed child? And if you would do the Punnett square, similar to what's up here, you would come up with a 50% chance. And those definitions of homozygous and heterozygous are on there too, just for your recognition of traits. So I have some basic, bigger picture comments down here. And again, we won't spend a lot of time on this, but feel free to look at this. And there's some things that, we look at evolutionary relationships with DNA. You can see that, humans share more than 98% of genetics with chimpanzees. We also really start to look a little bit more about sequences between and among individuals by DNA profiling. We've talked a little bit more about this in the past. We look at how we use DNA in forensics in natural disasters to identify victims. We look at analyzing food to determine authenticity of things and gourmet items and items like that. We can also say DNA analysis is also sort of like a time machine. We've heard a lot about using that on, as we said, forensics, criminal analysis. They've done analysis of DNA in, tombs of mummies, and you can see King Tut. We've looked at DNA and Thomas Jefferson and children, and fathering children and things like that. So again, it is sort of a time machine as we look back to that.